We're switching gears, so as my reminder, if you haven't taken the bacteria exam, that's due tonight. And we're switching gears, so we went over a ton of the most common bacteria that cause disease, and now we're flipping to all the viruses that cause disease, or the most common ones. Again, just like bacteria, we can't go over every bacteria. I mean, we would have to be an entire just bacteria class to get through all the bacteria and all the diseases that they can cause. We just go over the top bacteria that you're most likely to encounter in your careers. Same thing with viruses. We're going to go over the most common viruses that you will probably experience, or you either have experienced or will experience at some point in your life. So in this PowerPoint, we're not going to start getting into the actual viruses and the diseases they cause. That will start Wednesday. But it's a little bit just a background on viruses, just like we had entire units on bacteria and how do they reproduce and their genetics and all that stuff. We really haven't had any background on viruses yet. So it's all about just what are viruses, what are they made of, how do they reproduce, how do they get into cells, things like that. And then we have a little blurb on what are called prions. And so we'll see how far we get to today. I wrote myself a note. I can get through this in 65 minutes. We'll see. Whatever we don't get through, I'll record and put out there. Chapter 6. I know, I saw that. I'm like, oh, it's not chapter 13. That was the old textbook version. It's chapter 6, which is nowhere near in the book where the actual virus diseases are. It's kind of at the beginning, just kind of basics of viruses. So in this PowerPoint, we're going to go through just some characteristics of different viruses. How do they replicate? How they can cause cancer? If you want to grow a virus, how can you do that? We have one slide on, are viruses alive? And then we have a brief discussion on prions as well, which I'd like to get to because just the timing of the year. So some basics of viruses themselves, and I just threw some stuff on there so I didn't have to write it on the board. One, viruses are extremely small, which means we didn't even know that these existed for a really long time. And that's because we had microscopes finally and we could see bacteria. Well, viruses are even smaller than that. So even with our, awesome microscopes, a lot of times we still couldn't even see these viruses. Just showing in comparison some different cells. So here is a staph bacteria. Again, we've seen those already in our lab. We magnify them 1,000 times. Well, that's a virus compared to the size of a bacteria. So super tiny. I mean, that's one of our smaller viruses, but super tiny. Super tiny, that we wouldn't even realize it was anything. It could have just been a spot on the slide, a little piece of dust next to the bacteria. So they're super small. They are acellular, and anytime you put a at the beginning of something, it means it's not or without. So it's not an actual cell. It does not have a cell membrane. It does not have organelles. It does not have that cytosol fluid. It's not a cell. It does have genetic material, and it will have either DNA or RNA for its genetic material. So that's why on Wednesday we're going to talk about just the viruses that cause, the top viruses that cause disease that have DNA. And then we spend next week 2 full lectures talking about the viruses that have RNA as their genetic material. It has either or. They can cause tons and tons of different types of infections and diseases. They do not do any type of metabolism on their own. They need a host to be able to do that. They can't respond to the environment whatsoever. They can reproduce, but not on their own. They have to get inside of a cell, and then they use the cell's metabolic pathways. And viruses are always found in one of 2 different states, an extracellular state or an intracellular state, meaning we find the virus outside of a cell or inside of a cell. Whether it's out there trying to be infective or when it's gotten inside of a cell and has hijacked it. And it's because of the 2 different states, it's like 2 different immune system responses. So if a cell or a virus, if a virus is found in the extracellular state, so it has not gotten inside of a cell yet, we call it a virion. So it's like a virus, but it's just not inside of a cell yet. It's an infectious organism at that point. It's trying to find a cell to hijack. But when it's in the extracellular state, it is made up of 2 things. A capsid, which is a protective coat on the outside, and the nucleic acid. Whether it's DNA or RNA, that's the NA, the nucleic acid of DNA or RNA. So it's made up of genetic material and a protective coat that's there just to protect the genetic material. And because it's made up of both a capsid and nucleic acid, we sometimes call this structure a nucleocapsid. We just smush the two words together. It's great. Some viruses will also have a membrane or an envelope on the outside of them. They don't make them, they steal them from whatever host that they were released from. So some, and we'll talk about it when we get into viruses, when we talk about enveloped viruses, some can have a little phospholipid envelope on the outside. Because they are extracellular and not inside of our cells yet, the humoral immune system that recognizes extracellular pathogens would be triggered to recognize these and fight them off. Virus finds a cell, it can get rid of the capsid. So once it gets inside of the cell, it doesn't need that protective coat anymore. The job of that virus is to get that genetic material inside of the cell, and then it will hijack it with the genetic information that it has on that nucleic acid. And so in the intracellular state, it gets rid of the capsid, just as the nucleic acid found. And once it's inside the cell, now our intracellular or cell-mediated immune response comes into play. Our T cells are going to have to recognize that this is an infected cell, and it's going to have to kill the cell. It's different types of immune responses. Now, as I said before, genetic material can vary because some viruses are DNA and some viruses are RNA. And so there is more variety in viruses when it comes to genetic material than any other cell out there. These aren't even cells, but they have more genetic variety. And because they have so much genetic variety, we use, what do they have? DNA or RNA to help categorize them and classify them. Which is why we have an entire PowerPoint in DNA viruses, and we have an entire PowerPoint in RNA viruses. And then even knowing whether they have DNA or RNA, there's also some variation, because if you have DNA, you could have double-stranded DNA, which is pretty normal. You could have single-stranded DNA. It's pretty rare, but some viruses have it. If you're RNA, you could have double-stranded RNA, super rare. Some viruses have it, or you can have single-stranded RNA. And then even of the single-stranded RNA, it varies on, like, well, which strand do you actually have? Some RNA is also linear in little segments. The flu virus has a bunch of, several segments of genetic material. And some viruses just have one single circular band of genetic material. So we use whether is it linear and segmented or singular and circular to help organize them, but they don't have a lot of genetic material. So the amount of DNA or RNA they have is still a lot smaller than any kind of actual cell out there. So this is showing you the genome, the DNA of E. coli. So all the little squiggles, that's all the DNA of E. coli. And it's not even showing all of it. This is the DNA of a virus. I mean, there's a lot less. Again, they don't have to have the DNA to make organelles and do metabolism or anything on their own. They just need the DNA, have to have the DNA on how to hijack a cell and have the cell do all of it. But just because they don't have a lot of DNA doesn't mean they cause some very deadly, very catastrophic diseases. So viral hosts, so depending on what cells, what organisms viruses can get into, viruses are usually pretty specific for what cell they get into, and not even what cell, what host they can get into, and then what cell of that host. So there are plant viruses that only infect very specific plants, which means if you eat this plant, are you going to get infected with the tobacco mosaic virus? No. They're very specific for their host, and they're very specific even for the cell inside of a host. Just an example, HIV only affects humans. And of humans, it only affects a very specific cell. It only affects T cells. And of the T cells that we have, because there's cytotoxic T cells and regulatory T cells and helper T cells, it only affects just T helper cells. So some viruses are very, very, very specific for what cell in what host. Now, one unique group of viruses infects bacteria, And they're called bacteriophages because we're going to talk about, they're just this whole classification of viruses all in itself. We got a lot of bacteria out there. We've already talked about them. There's a whole group of viruses that all they do is infect bacteria. Again, a virus needs to find a cell. Well, bacteria is a cell. There's lots of cells when bacteria get in and cause infection. So you can have lots of viruses that are all trying to hijack that one bacteria. So bacteriophages, very common, and we usually will just call them phage viruses, and we just leave off the word bacteria. So if you ever hear anywhere anything that talks about a phage virus, just know, that's a virus that infects bacteria, which means it's not going to infect us. It's not going to cause us issues. But I mentioned in lab when we were testing everyone for Staph aureus, there are viruses that are specific for Staph aureus, because I've tested positive for a virus that infects Staph aureus. And if a virus infects Staph aureus, it can kill the bacteria, which makes me really likely to never have a Staph aureus infection. Now, this again is just showing you some size comparison of how small some of these viruses are. So this is showing an E. coli bacteria compared to the size of a red blood cell. So bacteria, very small. Red blood cells are very small, bacteria even smaller. If we zoomed in on that E. coli, we can start to see the size of different viruses. Bacteriophages, I think even though they don't affect us, they're like the coolest shaped bacteria. I mean, they look like something, or viruses, they look like something that should be landing on the moon. And these are just viruses that infect bacteria. I was going to say that some of them have very unique shapes and structures, and also a big variety in size. A smallpox virus is one of the biggest viruses, but it's still extremely tiny. That's the biggest virus we got, and it's still extremely tiny next to an E. coli. The polio virus, which is making a comeback, is one of the tiniest viruses. And it's like you can almost not even see the tiny, tiny, tiny speck that it is up there. Now, as I mentioned before, bacteria, or viruses, I keep saying bacteria, viruses, when they are outside of the cell, do have a protective capsid. It's made up of little protein units called capsomeres. So all of these little tan, kind of circular structures are all individual little protein capsomeres. And they just come together to form that nice protein coat. Now, its main purpose is for protection. That genetic material is extremely important. That's what the whole virus is based on. So it's really there to provide protection, but it also helps it attach so that these viruses can attach to very specific things on different cells so that it knows what cell to infect. And the capsid also gives it a lot of new, cool, unique shapes. So scoot back a second. These different shapes are all because of that outer capsid. These are just some of the different shapes, something that looks like it could be landing on the moon. If I go back, it could have that kind of icosahedral shape to it. Some other unique shapes, that long, like that tobacco mosaic, a long tube, more circular. There's one we'll get to that even has a bullet shape to it. All the shapes are depending on just that capsid. So the capsid there protects, attach, but also gives it cool shapes that helps us identify it. As I also mentioned before, some viruses have an envelope. They don't make them, they steal them. When they get released from a cell, our cells all have a membrane on the outside, and it steals some of it. So it's still that same phospholipid bilayer that our cells have. They just steal some of it as they get released from the cell. Now, as they release and steal it, that virus itself, when it was living inside of the cell, that genetic material coded for making certain type of proteins. And those proteins stick on the outside envelope or membrane, so that it's these glycoproteins that recognize cells in the body. It's also these glycoproteins that our immune system has to recognize as foreign. When COVID came out, again, that was some of the first things they had to figure out. What are the glycoproteins and what cells are they sticking to in the body? And does COVID affect just one type of cell in the body? What do you think? No, and that became an issue is because some people lost their sense of smell, but not everyone did. Some people, they had a cough or they had a sore throat. Some had digestive issues. It caused such a huge, wide variety of different types of symptoms. And some of the symptoms were just because of the immune system response, not because of the virus, but some were virus-driven. And that's just because the glycoproteins, they stuck to a specific type of receptor. But then unfortunately, that receptor is found on lots of different cells in our body, which cause lots of different issues and lots of different symptoms for it. This is just showing another example that there's an envelope on the outside, but it's got a different shaped capsid. Now, viral replication. So the virus has to recognize a cell, it has to attach to it, it has to get inside of it, and it has to hijack that host cell to start making more of that virus. Now, we're going to go through all the steps that a virus does to hijack a host cell and reproduce and make more for the viruses that infect bacteria. And then we're going to do a comparison to the viruses that infect us and other types of animals. Similarities, lots of similarities, but then we'll just talk about some of the differences. So viruses that hijack bacteria. Again, they cannot reproduce on their own. They have to hijack some type of bacteria. And they go through what's known as lytic replication. Now, lysis is where the word lytic comes from. Anything that ends in lysis, like hydrolysis, think of other words, hemolysis. Lysis means to break apart. And so lytic replication tells you what's going to happen to the end. Is that host is eventually going to break apart. It's going to die. So that's why they call it lytic replication. So it goes through 5 stages. It first has to attach. I don't remember if I had little things come up. It first has to attach. And again, it's going to recognize a very specific bacteria for it to attach to. And again, these bacteriophages have some really cool little structures to it. They look like little moon organisms. So they first attach to their host cell. And then you have the entry. And again, the only thing that gets inside of the cell, or the only thing that's required to get inside of the cell, is just the genetic material, whether it's DNA or RNA. So you have the entry. Then you have the synthesis. Synthesis means to make. It's at this stage, this poor little bacteria has been hijacked, and it has now become a virus-producing factory. And the only 2 things that a virus is made up of is the nucleic acid, and proteins. That protein coat, that capsid on the outside. And so this bacteria is making the genetic material, the little blue squiggles, and it's making all the little protein parts that will become part of the actual capsid. So it just starts to make everything. Once it makes everything, then you assemble it all. So now it actually looks like viruses, and then you have release of the viruses, and then they will go out and infect more bacteria. So it first has to attach, get the genetic material in, make parts, assemble the parts, and then release them to go do it all over again. Again, just another picture of it where you have the attachment, the entry of the genetic material, the synthesis, making all the parts, assembling all the parts, and then you have the release of viruses to go hijack more. Some viruses can go through what's also known as lysogeny, or the lysogenic cycle. It's a modified life cycle. It means they go through that lytic cycle, but then they kind of just pause for a little bit. And so this is showing an example. This particular virus, it attached, it got entry of the genetic material in there, but now this bacteria that has viral genetic material in it doesn't become a virus-producing factory yet. And bacteria are going to do what bacteria like to do. They're going to reproduce. But this bacteria is going to reproduce, and it's going to reproduce not just its own bacteria DNA, it's going to reproduce some of that viral DNA as well. So now instead of having 1 bacteria with viral DNA, you have 2 bacteria with viral DNA. And those 2 bacteria will reproduce, and now you have 4 bacteria with viral DNA. And it can do that for several generations of bacteria as they're growing and dividing. And at some point, something will trigger them to go back into the lytic cycle. And they're going to take that viral DNA and make all the parts and assemble all the parts. And you've got lots and lots of bacteria producing lots and lots of viruses all at once. So it's a modified life cycle. It's like the normal life cycle with a little bit of like pause where the bacteria are just going to reproduce. Now, when they're just reproducing and not making active viruses, we say they're in a temperate phage, or also known as a prophage. Different books call them like the same interchangeable words. They're just an inactive virus. The virus is not reproducing more viruses at this point. What can happen, though, is that while these bacteria are harboring viral DNA, the bacteria can change a little bit. You could have a bacteria that, has never made a flagella before. Well, maybe all of a sudden it does. And it's because on that information, that little bit of DNA on the virus is like how to make a flagella. Or it'll change a little bit of shape on the outside. So sometimes the bacteria change a little bit when they are hijacked. It's just known as lysogenic conversion. Bacteria just change. They're infected. So this looks a lot. This is the normal lytic cycle. I say focus on this first. Where the virus attaches, it gets the genetic material in, makes all the parts, they assemble all the parts, it gets released. Normal lytic cycle. That lysogenic is the genetic material got in, but before it starts making more viruses, it just becomes part of that bacteria DNA, and the bacteria reproduce like they always do, and it reproduces some of the viral DNA as well as the bacterial DNA, and now you got 4 bacteria, all of them are infected with the virus, the genetic material, and at some point, something trigger them, it's called induction, to go back into the lytic cycle. And then they start to make all the parts and become huge, mass-producing virus factories. Where I think it keeps repeating. The comparison of a lytic cycle to a lysogenic cycle. So lytic cycle, they get in, hijack, cell dies right away. In the lysogenic cycle, bacteria, there we go. The bacteria uptakes the viral DNA, and the bacteria just reproduces. And every new bacteria now still has viral DNA in it. And then, yes, it waits until some trigger causes it back to go into the lytic cycle. Another just showing the differences. Now, animal viruses. So these are going to be viruses that infect us. Have the same 5 basic steps. They still have to attach. They still have to get the genetic material in. They still have to make the parts. They still have to assemble them, and they still have to release them. But then there's some differences of how they do all of that in each of the 5 steps, which is why we're going to go over the differences. It's pretty simple when a bacteriophage infects bacteria. Some of the differences are a good number of viruses that infect our cells, any animal, have envelopes. They have that outer membrane that they stole. Our cells are eukaryotic, which just means our cells have a full nucleus inside of it, where our DNA is hiding out, unlike a bacteria where its DNA is just floating around in the cell. And none of our cells have cell walls, as bacteria do. So there are different kinds of cells that have to get hijacked. And so there's some slight differences in what happens. So the first thing that still has to happen, we still have to attach. That virus still has to attach to our cells. There's usually some type of chemical given off by our cells that attract the virus to that specific cells. The viruses that infect us don't have tails or tail fibers, which means they don't look like they have landing gear. So if I go back to these little things are called tails or tail fibers. So the things that look like they should be landing on the moon only infect bacteria. Our viruses that infect us don't have those cool looking landing gear tail fibers. Instead, the viruses that infect us use some type of little glycoprotein spike. usually found in that outer membrane, to attach to our cells. A little different in attaching. They still go through that second part. They still have to enter. Oh, there was the tag. So 3 different ways that it can go through entry. The first is called direct penetration. It attaches to one of our cells, and then it just injects its genetic material in. There we go. Now all of a sudden our cell now has genetic material. That's one way I can get the genetic material in. It's called membrane fusion. So if this is a virus, I don't need that. If this is a virus and it has an envelope, and an envelope is made up of a phospholipid bilayer, our cells are a phospholipid bilayer. When they attach, all that phospholipid bilayer just kind of melds together, and the entire virus, capsid and genetic material, goes in. So it's called membrane fusion. That envelope fuses with our membrane, and the entire virus, the whole nucleocapsid, goes in. Again, it does not need that capsid on the outside, so the first thing we'll do is get rid of it. But it uses that whole envelope to just kind of meld and fuse together to get the virus inside. And the third way it can get inside of our cells is through endocytosis. It's almost like a phagocytosis, where our cells, the whole membrane goes up and engulfs the entire virus, envelope and all, and brings the entire thing inside the cell. Again, it doesn't need all that stuff. It just needs to get that DNA out. So it's going to get rid of the envelope, it's going to get rid of the capsid, and what you're left with is that genetic material. So it's either going to inject the DNA, the whole membrane will fuse, kind of meld together, or it just literally engulfs and brings in the entire thing. So there's different ways, different methods for the virus to get inside of our cell. It'll go through the third step. It still has to go through the synthesis. It still has to make the proteins. It still has to make that nucleic acid, whether it's DNA or RNA. I'm going to draw stuff. Strategies on making DNA or RNA, because some viruses are made up of DNA and some viruses are made up of RNA. And as we already went through this semester, we can do DNA replication. But we can also make RNA as well. That's the transcription. But it also makes a difference. Does the virus have double-stranded DNA or single-stranded DNA? Or does it have double-stranded RNA or single-stranded RNA? That makes a difference when we're starting to assemble all of this, or not even assemble, start to make it. If we're going to make new viruses, we have to make sure we make the same kind of genetic material they have. If it is a DNA virus, all the new DNA is assembled in the nucleus. That's where our DNA is assembled and is where it's found. If it's an RNA virus, all that new RNA is made out in the cytoplasm. That's where RNA goes. Now, the RNA viruses, they're the most confusing because there's 3 different types of RNA that a virus can have. So it can have double-stranded RNA. It can also have single-stranded RNA, but there's 2 kinds of single-stranded RNA. One's called a positive sense, and one is called a negative sense. I'm trying to think of the best way. I want to draw it out yet. The difference is, so when we go through, no, I'm just going to go it. When we go through transcription, make RNA, or messenger RNA. This is our cells. First, our DNA, is it single-stranded or double-stranded? Double-stranded, always. RNA that it makes, is it single-stranded or double-stranded? It's single-stranded. We went from a double-stranded DNA, and all of a sudden we just have one strand of RNA. We had to copy one strand of DNA. So if this, if this was an A and that was a T, all right, well, if that was an A, it would pair up with a U. But if this was a T, it would have paired up with an A. The biggest question is, well, which strand are you copying to make that single-stranded RNA? And that's why we get into, there are two different kinds of single-stranded RNA. One copies one side, one is a copy of the other side. And it does make a difference on which one it is because of the information that's found on it. So that's just kind of why we've got two different kinds of single-stranded RNA. Because it came from double-stranded DNA. So if a virus has what's known as the positive sense single-stranded RNA, I say this is going to be as close as you can get as your messenger RNA. So messenger RNA is going to be as close as you get to single-stranded positive sense, single-stranded RNA. It's almost like it copied the right side. It knew which side to copy. And if we want to make more viruses, not that we want to, but our cells are hijacked to do that, if we want to make more positive sense single-stranded RNA, I'm going to use different colors. I'm going to use the red because it uses red up there. This is viral DNA. And you need to make more of it. We'll just say we have, I don't know, an A, a U, and a C for whatever reason. It's picky letters. And I want to make more of it. First, make a template. Because our cells don't know, oh, I see an A, I should bring in an A. They don't see something that they have to make exactly. They have to copy a template. So they have to make the opposite strand. And what is the opposite? This is not going to erase well. What is the opposite of A in RNA? What would pair with an A? A U. What would pair with a U? An A. And what would pair with a C? A G. It has to make a template. And from that template, our cells are like, oh, every time I see a U, an A, and a G, it knows to bring in an AUC, and it will mass produce that RNA. Because it's making an opposite strand as the template, what's the opposite of positive? Negative. This template is the negative strand of RNA. So that's why on this picture, once that single-stranded RNA gets inside the cell, the very first thing our cell will do is make a template. It makes the opposite. And from that template, our cells now become mass-producing factories of that positive sense single-stranded RNA. And you can make lots and lots of copies. But the other thing our cells have to still make, other than genetic material, is proteins. What organelle makes proteins? The ribosomes. Ribosomes can recognize the positive sense single-stranded RNA, and it will go straight into translation and start churning out proteins. And so a ribosome's gonna be like, oh, yep, I know how to recognize that. I can read it just like normal translation. It reads it 3 letters at a time, bringing in all the correct amino acids. It makes all the different viral proteins. The positive sense single-stranded RNA is gonna be most similar to messenger RNA. Because our ribosomes recognize messenger RNA, it recognizes the positive strand, and it will just sit there and mass produce proteins. Negative sense. If a virus has the opposite strand, it doesn't have the positive strand. A ribosome cannot recognize the negative strand. And so the very first thing that cell is going to have to do, one, it does still have to make negative sense single-stranded RNAs, and to be able to make a negative strand, we have to make a template. And what's the opposite of negative? Positive. And so the cell will first make the template, which is going to be the positive strand. And it's this template that the ribosome recognizes. So the ribosome does not recognize the original genetic material because it's negative strand. It only has to recognize the template. and it's from that template, it will sit there and mass produce more negative strand. So it's very similar, we just kind of flip it around a little bit, and ribosomes cannot recognize that initial viral genetic material. And so until it makes that template, the virus can't reproduce. Of RNA viruses that have double strand. I shouldn't say a whole group, there's not very many, but there's some. Again, they're a virus that has both. They have both the negative strand and the positive strand. And so the very first thing that will happen once it gets inside of our cell is it unwinds the two strands. So we've got our negative and we've got our positive. And that positive sense is going to, again, act just like messenger RNA. A ribosome is going to be able to recognize it. It's going to be able to make all the proteins that it possibly needs. And it uses each other as the templates. So if we need to make more of the negative strand, it reads the positive. If we need to make more positive strands, it reads the negative, which is nice because a double-stranded RNA has both strands. It already has those templates made for it. And so it can easily make more genetic material. And it already has the positive strand as one of the strands, and so it can easily, that ribosome can easily read it and make proteins. So there's a lot of difference in the synthesis part depending on what kind of genetic material it has. And then it still goes through the last two stages. It still has to assemble all the parts that it just got done making, made all the proteins, made all that nucleic acid. It still has to assemble them and release them. Picture got too big. So if it's a DNA virus, all the assembly takes place in the nucleus because that's where the DNA is found and where it was made. If it's an RNA virus, all of the assembly takes place in the cytoplasm because that's where RNA is made and formed and read. The number of viruses that get assembled depends on, one, the type of virus it is, and two, the health of the host cell. If you have a very healthy cell, it hasn't been around for a long time, it's not on like its last day before it dies, it's going to make and assemble more viruses. And some viruses, depending on what type it is, mass produce faster and easier inside of certain cells. Now when it gets released, if it's an enveloped virus, as this one is showing, so it assembled the entire virus, it's got its nucleic acid, it's got its capsid, but when it leaves the host cell, it steals that envelope. It steals some of that phospholipid bilayer. And on its way out as well, it also makes sure that it takes some of this viral glycoprotein that the cell was making while it was hijacked. And so you can see that it is leaving the host cell, stealing some of that host cell outer membrane. Now, as it's leaving the cell, is it killing it? Oh, it's not actually killing it, which is actually a great strategy for viruses. If our cells can become virus-producing factories and it doesn't kill the virus, or it doesn't kill the cell is always a virus-producing factory. Why kill the assembly area? And so a lot of viruses don't kill the cells. At some point, something can trigger it that when they become released, the cells will die. But a lot of enveloped viruses don't kill the cells, at least not right away. Naked viruses that are not stealing the envelope, they are usually released by cell lysis or bursting forward. Oh, I didn't have a picture. Also known as exocytosis too, where they literally leave the cell, they burst open. Those are generally going to kill the cell. But envelope viruses are usually a little more persistent. They usually hang out in the body for a longer period of time before eventually the immune system can get rid of them, but it's not even a guarantee that it ever will. Now, because some viruses are persistent, they don't kill the cell, and because some viruses can go dormant, they can just stay inside of our host cell, and our host cell, although it's infected, isn't actually doing anything about it. There are viruses that can go dormant. So the virus is in the body right now, but it's not doing anything. So they're called latent viruses or proviruses, and not all viruses can do this. There's one very unique group that I think we go through on Wednesday, that is the herpes group of viruses that is known for going dormant. And everyone always thinks of herpes as just one kind of herpes. It's like, oh, cold sores. There's so many herpes that we'll go through on Wednesday, not just the cold sores. And it's like almost a guarantee that one of you has been infected with one of the herpes. And I'll admit, I have. The question is, you gotta wait till Wednesday to find out which one. So a lot of the herpes viruses, they incorporate part of their DNA into our DNA, and then it just hangs out. That virus will cause it to go active. This is one of the reasons why all of a sudden I'm like, cold sores start to appear. The virus is in the body forever. If you've ever had a cold sore, you have one of the herpes viruses. It's just hanging out in your body. And then something will cause it to be active, and that's when you start having the cold sores, the symptoms of the virus reproducing and causing death of cells. But another one, if you've ever had the chickenpox, it's in the body, and it can reactivate as shingles later on. Same virus, just causes different disorders. Another virus that almost always incorporates itself and hangs out in the body is HIV as well. Like once you have HIV, you don't get rid of it. Your body doesn't recognize it. Your immune system is under attack, and it just becomes part of the body. Our goal these days with our drugs is to keep it dormant. Just showing a little comparison on viruses that infect bacteria versus animal viruses, going through those attachment, penetration, synthesis, assembly, release. Just some differences between them. Is that we've realized now is that viruses can cause cancer. Not all of them, but viruses can cause cancer. Now, a little bit of background on cancer. We have a whole cell cycle that tells our cells to reproduce and replicate and go through mitosis, which is supposed to be. But we have a whole bunch of checkpoints that say, okay, well, we're done now. We don't need any more cells, and we're fine. What ends up happening in cancer is that somewhere along the line, one of those checkpoints that says, we need to stop mitosis goes wrong. And so these are the cells of the M is mitosis. And normal cell cycle where your cells are, they're growing. They'll eventually start making more DNA. They'll eventually go through mitosis and grow and divide. But we have all these checkpoints along the way. So we have genes for cell division that we can turn off so that we're like, okay, we don't want to replicate. We have genes that stop cell division that we can then also turn on as another checkpoint. We're like, all right, we've got all these different genes that say whether cells can replicate or not. Cancer is when something's gone wrong with those genes, where you can no longer stop cells from dividing. And cells will divide, and they will divide, and they will divide, and no checkpoint is going to tell them to stop. That's what cancer is. It's uncontrolled cell division. Now, it's called neoplasia. It's also known as new growth, but it's uncontrolled cell division. And because there are no checkpoints anymore that the cells need to stop, there's also nothing to check to see if there are abnormalities to those cells. And so now you have cells that are not normal, which would have been triggered to not divide because they're abnormal, but nothing tells them to stop dividing. And so now you have abnormal cells that are growing and growing and dividing, and nothing's telling them to stop. That develops into a tumor. It's uncontrolled cell growth. Nothing tells it to stop, and the cells are not normal. Now, if those cells remain in the same place, we say you have a benign tumor. If they start to spread throughout the body, you have a malignant tumor. If you had to pick a tumor, which one would you want, a benign or malignant? You'd want a benign. That just means the cells were growing uncontrolled, nothing caused them to stop, but they stayed there, which means you can easily go in and surgically remove that mass of cells. Solved. They start to spread. Now you have cells that nothing is telling them to stop growing, and they're traveling anywhere, potentially anywhere in the body, and you can have tumors everywhere, and none of these cells are growing normally. They're abnormal cells, and they're going to interfere with just about anywhere in the body. It's bad. And it's a lot harder to treat. You can't just remove it. Now, terms on here, and I'm going to draw this out. This is like our normal DNA. So we have what's known as a proto-oncogene. Anytime you hear the word onc, what do you think of? Like oncology. That's what the oncology unit at Gunderson is. It's cancer. Anything that has pro or proto in it, it means it's inactive. It's not doing anything anymore. And so a proto-oncogene, this is a gene that can cause cells to divide, cancer, but it's inactive because it's not doing anything. And so this is our normal DNA, and it's got genes. Genes are just snippets of DNA. And it's got a gene, but our cells are not going to divide. It also has a gene that's going to repress or stop cell division. And so at this stage, our cells are the cells going to divide? It's got an oncogene that's not doing anything. Cells aren't going to divide. It has a repressor gene that's going to stop cell division. Cells are not going to divide. Now, what can happen, though, is when your cells get infected with a virus, the only thing that really gets inside your cell is the cell's DNA. If the cell or the virus's DNA lands right here next to the proto-oncogene, it can turn that proto-oncogene on, and now it's just called an oncogene. So now you have a gene that's going to cause this cell to divide. Is that good or bad for cancer, for cells to divide? For us. Bad for us. Our genes to the point that our DNA is like, oh, I guess our cells got to start dividing. But luckily, we still have a gene right here that's like, nope, not going to divide. So at this stage, is the cell going to divide? No. But you've activated an oncogene, that it's like, oh, it's just ready. It's wanting the cell to divide. But what can happen, too, is that virus can either, one, insert in two places, or two, you could have two viruses that have infected your body. You have one virus that it got into the cell that activated that oncogene, and if you have another virus that inserts itself anywhere right in the middle of that gene, is that gene going to be able to do anything anymore? No. And so it can't stop cell division. So this is going to say, let's cause the cell to divide. And this is like, I got nothing to say about it. The cell is going to divide. Is that good or bad for us when it comes to cancer? Bad. So this is how viruses can cause cancer. Viruses get in and they incorporate into our DNA. And if they incorporate it into our DNA anywhere near these genes that are there to stop cells from dividing, they mess with them, and then our cells are going to start to divide. Now, there are other things that can cause cancer as well. It's not just viruses. Other things that can cause cancer, UV light can cause cancer, radiation, anything that's called a carcinogen. But viruses can cause cancers as well. They say about 20 to 25% of all human cancers are caused by viruses. That's a lot. Specific ones that I put up here, these are not the only ones. Burkitt's lymphoma, we'll get to these as we get into these different viruses. Burkitt's lymphoma is caused by the Epstein-Barr virus. Hodgkin's disease is also caused by the Epstein-Barr virus. Kaposi's sarcoma is caused by HIV. And cervical cancer is caused by HPV. And it's not the only cancer that HPV can cause, but it's one of the more common cancers. Now, HPV has become kind of like our, most famous virus, because what do we have for this virus now that can prevent an HPV infection? Didn't have it back in my day. We have a vaccine. We have the Gardasil vaccine that is specific. It is a vaccine to prevent this virus infection, which then can prevent cervical cancer. Other things can cause cervical cancer other than just that. So to say you're vaccinated against this and say you'd never get this, you would just never get cervical cancer caused by HPV. So if we can develop vaccines for viruses, we can actually develop vaccines against cancer. It'd be great. Again, if we can develop enough vaccines to target all the viruses that are linked to certain cancers, we could potentially stop 20 to 25% of all human cancers. That's huge. And that's where a lot of research is going. I mean, it's pretty impressive. You know, when I was younger, we did not have a vaccine for HPV. Now, my daughter already got the first one. You know, it's like, yay! We have vaccines that can prevent cancers. Your very own virus. There are 3 different types of media that we can use. One is these are still viruses. For them to grow, they still have to get inside of a cell. And so we can use mature organisms. If we want to grow bacteriophages, we can first grow the bacteria, then we can infect those, the bacteria with the virus, and the bacteria become virus-producing factories. We can use plants and animals as well. Same way, we know that those viruses have to get inside of those cells. This is showing an example how they made a lawn of growth. So they uncovered the entire plate with bacteria, just like what you guys did upstairs. But they also put some viruses as well on that plate. And anywhere you can see bacteria not growing, the little black spots, that's where viruses got in and were getting reproduced, and they were killing the bacteria through lysis. We can also use embryonic chicken eggs. Again, an egg is one cell. And because it is such a big cell, we can mass produce viruses in that one cell. And it's also one of the top reasons we use it in mass producing vaccines, is because we can put those viruses in the cell, mass produce them, but we can also make sure that that virus is in altered form, that it can't cause disease. And the more common way of growing up viruses in a lab is using some type of cell or tissue culture. Looks just like this. We've got cells, we put them in a nice nutrient broth, so it's always a reddish color, so that the cells are happy, and then we put the virus in with them. And as the virus gets in and infects all the cells, it will eventually start to kill the cells, but they become huge virus-producing factories. So different ways we can grow viruses. Now, my question, are viruses alive? And we talked about this like way at the beginning, I think, in this semester. No, but there's still like a resounding argument about it. So some individuals will say they are just complex pathogenic chemicals. No, they're not basic characteristics of life. They can't respond to the environment. They can't reproduce on their own. They can't do metabolism on their own. They don't meet the same characteristics of life. They're just really complex, pathogenic, disease-causing chemicals. Other people will say, but come on, they are like the most complex, pretty genius things out there. They're so genius, how can you say that they're not alive? I mean, they have very sophisticated methods on recognizing cells and getting into cells. They hijack their host cell. Like, come on, how do you hijack something to not even be alive? And they do replicate themselves, just not on their own. And so the other part is, some will say, no, they're just pathogenic chemicals. Others are like, well, we'll just call them the least complex, because they're pretty simple, living entities out there. Them as they're alive, because I say, we will kill them. How do you kill something that's not alive? Killing them is kind of just saying you inactivate them. I don't know. They are pretty sophisticated for not being living organisms. Now, my last thing that we're going to get to, which I was happy to get to it, are to talk about prions. Prions are not viruses, but they get grouped with this chapter because of how simple they are. So they are still disease-causing, just like viruses are. They're not alive, just like viruses aren't technically alive. So a prion is an infectious protein. And that's where it got its name. It's just protein that's infectious. And we called it a prion. Now, it's a protein, and proteins usually have some structure where they've got these little helical shapes to it, or they have these long, straight sheets. Now, in mammals, that includes us, in our nervous system, because that's what these prions seem to target, in our nervous system, our protein P, so it's our little protein P, has a whole bunch of these little helical shapes to it. This is the normal shape of protein P in our nervous system. But what can happen is that if you get a prion, it's still a protein P, but it's got a different shape to it. Now, what do we call it when you change the shape of a protein? Denature it. When you change the shape of a protein, can it do its job? No. So that's bad. So this is called a protein. It's a prion protein P. This is the protein P. It's still a protein P, but it's a slightly different shape, which means it cannot do its job. Now, we call it infectious for a reason, because once this protein P with this misshape gets into the nervous system, it will cause all of these little helical shapes to unwind. It's almost like, hey, it's not cool anymore to have a helical shape. You should really unwind and have these straight, they're called beta sheets. Our own protein shape will start to unwind. And that's why they're called infectious. It's a protein that encourages other proteins to change shape. So our normal cell protein P becomes an infectious, misshapen protein P. This only happens in the nervous system. So it's just a protein. So there's no nucleic acid or anything else. It's just an infectious protein. Now, because it gets into the nervous system and our protein P in our nervous system changes shape and it can't do its job, we start to lose tissue in our nervous system. And it will form something known as spongiform encephalopathy, which is a huge long word, but I like to break it down and it tells you exactly what it is. Pathy means disease, encephalo means brain, and so it's a disease of the brain where it looks like a sponge. So it's spongiform encephalopathy, you're like, oh, that sounds horrible. It's just a disease of the brain that looks like a sponge. So brain tissue should not normally have all these big white gaps everywhere. It shouldn't have these little vacuoles of empty space. You have tissue dying because the protein P is not its normal shape. Now, one of them, and it's on my next slide too, is bovine spongiform encephalopathy. Bovine spongiform encephalopathy comes from a specific animal. What animal? Cows. What is this also known as? Mad cow disease. Now, it was infected in cows. If a cow's brain tissue is starting to look like this, is the cow acting normal? No, hence mad cow disease. They were acting crazy. It's because they were losing brain tissue. The unfortunate part is that people that were eating infected tissue were also getting it from the cows, which is why if you ever donate blood or plasma, I think they always ask, like, did you ever travel to, Europe during these particular years? Because there was like a huge outbreak in Europe and England of mad cow disease. Now, it's transmitted by ingestion or contact with the mucous membranes. Ooh, this is also called variant Creutzfeldt-Jakob disease. And you guess what CWD stands for? Chronic wasting disease, which is found in what animal? Deer, which is why I wanted to get to it because it's deer season. We run into an issue is that this is really hard to destroy. It's not alive, it's just a misshapen protein. So how do you kill a misshapen protein? You can't just I'll just autoclave it, because even autoclaving won't kill it. This would still, because it's not really alive anyways, but it still wouldn't kill it. It would still be infectious and that protein can still get in and cause issues. It actually has to get autoclaved at a higher temperature, and tissue has to be sitting in a really strong acid or a base while in the autoclave to eventually destroy it enough that it can't be infectious. This is just showing what normal brain tissue looks like, what spongiform encephalopathy looks like. And these are some of the different diseases that are caused by prion diseases. So the human prion diseases, that Creutzfeldt-Jakob, I want to try to say that, fatal, familia, insomnia, I don't know, and Kuru, an interesting part about Kuru, it's been eliminated. It was a prion disease. They only found it in like one African tribe. And it was very, very deadly. And then they realized what was happening is that in this particular African tribe, when someone died, it was customary to eat the brain tissue of that person that died. And then they realized like, oh, hey, no, it's causing more deaths. And then so they just were like, hey, you guys stop doing that. And they did. And so no one gets Kuru anymore. But different animal diseases. We know that bovine spongiform encephalopathy, or that mad cow disease, affected humans. So one of our biggest questions right now is chronic wasting disease affects deer. We see it all the time in the news, especially now that it gets to be hunting season. As deer like touch noses, because this can be spread by mucous membranes, they can be spreading it, which is why they don't want you to feed deer, because they don't want deer in large crowds together touching noses and mucous membranes. If mad cow disease could go from cow to human, can chronic wasting disease go from deer to human? You know, the problem is a lot of these prion diseases, you might not, I mean, it's a very slow, progressive nervous system disease that you might not even know you have this prion in your nervous system for 30 or 40 years. And then all of a sudden 30 or 40 years goes by and you develop it. How do you know for sure that it came from that one deer that you ate 30 or 40 years ago? We haven't gotten to the point, because chronic wasting disease is pretty new in deer. We haven't gotten to the point that we have humans that are even starting to get identified as having any type of prion disease. And even then, to say confirmation that it came exactly from a particular deer, I don't know. It's one of the main reasons they want you to get your deer tested for chronic wasting disease, is to just know like, hey, you should probably keep track of this. You know, if it was tested positive, maybe you don't want to take that risk that in 30 or 40 years you develop a prion disease and your brain tissue starts to look like a sponge. Or just know that if you still eat that deer meat, because that deer's still going to have nervous tissue running through the venison, that, yeah, you might develop something. And then in 30 or 40 years, if you are diagnosed with something, you can be like, well, that 1 year, my one deer did test positive. It was probably from it. I don't know. I still eat venison. I still love it. I guess I'm willing to take that risk. But we do still have our deer tested, just because we've yet to have a deer test positive. Knock on some wood. I don't know what we'll do if it does. So that was the end of my PowerPoint. I was glad I got to that part. Just on time. So things to do before I see you on Wednesday. So we did finish this PowerPoint, which means there is a quiz due